Cellular respiration involves oxidizing organic molecules like glucose and reducing oxygen, all for the purposes of producing ATP. And this all occurs in 3 stages. Now, in our previous video on glycolysis, we described how this starts with a molecule of glucose that undergoes a series of molecular rearrangements, at the end of which what we get out are 2 molecules of the high-energy electron carrier NADH, 2 molecules of ATP, and 2 molecules of pyruvate. Now, remember, it's ATP here that's going to be doing the work of the cell, and, well, frankly, 2 ATP isn't going to power much of anything. In fact, at this point, most of the potential energy from that glucose molecule is still to be found in those 2 pyruvates. And to get at that energy, what we need to do is move on to the next stage of cellular respiration, which we call pyruvate oxidation and the citric acid cycle. In a eukaryote, pyruvate oxidation and the citric acid cycle take place in the mitochondria, meaning that they can't begin until that pyruvate has been transported into the mitochondria. So it passes through the outer membrane through channels, and then it eventually crosses the inner membrane via a protein complex that is, sensibly enough, called the mitochondrial pyruvate carrier, or the MPC. And that transports the pyruvate into the interior compartment of the mitochondria that we call the matrix. Once the pyruvate has made it into the matrix, it immediately starts being processed. Now, there's really just a few key events that I want us to think about here in terms of what happens to these pyruvate. Firstly, one of the carbons is stripped away and is actually given off as a carboxyl group, or actually CO2 carbon dioxide that we actually breathe out. We also notice that that remaining 2-carbon compound is oxidized, and this results in the production of another molecule of NADH. And then finally, something called a coenzyme A joins to the remaining 2-carbon piece, and it generates what we call an acetyl coenzyme A, or as typically abbreviated, acetyl-CoA. Now, an important thing to remember here is remember that it's happening to 2 pyruvates. For each starting glucose, we had 2 pyruvate molecules. So actually, our input to pyruvate oxidation is 2 pyruvate, and our output is actually 2 CO2 molecules that we expire. It is 2 NADH, which we'll see at the very final stage of cellular respiration, and it is 2 acetyl-CoAs. It's those acetyl-CoA that are now going into the citric acid cycle. Now, just like with the glycolysis video, we're not going to get ourselves all worked up and caught up in the molecular details. I mean, they're pretty cool, but the fact is you can find them in any textbook if that's what you're interested in. Instead, what I want us to do is to think about the citric acid cycle with 3 questions in mind. Firstly, what happens to those acetyl-CoA molecules that enter the cycle? Secondly, what do we get out of the cycle as a result of this process? And then thirdly, what does this actually mean? What are the implications for ATP energy production? So to give you a bit of a hint here, the goal of the citric acid cycle is to complete the oxidation of what's left of that starting glucose molecule. Basically, electrons are going to be stripped away, taken by high-energy electron carriers off to the final stage of cellular respiration. So, let's take a look at this cycle and just highlight the key important events that we want to take away from it. Well, the first actually is when the acetyl-CoA enters the cycle because it's joined to a 4-carbon compound called oxaloacetate. And what that gives you is citric acid. Citric acid cycle, right? Okay. But what you're going to see as this cycle progresses is that that citric acid is going to be degraded back to oxaloacetate. And as that happens, some very cool events are going to occur. So let's just take a look at some of these. Notice that on 2 separate occasions, a molecule of CO2 is given off. We are going to breathe that out, but what that CO2 actually represents is the last of the carbons from our original glucose. You'll also notice that there's another event that results in the formation of a molecule of ATP. But super importantly, there are a series of oxidation reactions that result in the formation of 3 molecules of NADH and 1 molecule of another type of high-energy electron carrier called FADH2. So, if we think about what we put in, we have to remember, okay, that 2 acetyl-CoA went into the citric acid cycle. So what that actually means is our total output from the citric acid cycle is 4 carbon dioxides, 2 ATPs, 6 NADH, and 2 FADH. But let's take a whole other step back and think about where we started. We started with a molecule of glucose. That molecule of glucose has gone through glycolysis, and it has now gone through pyruvate oxidation and the citric acid cycle. And what have we got out at the end of this? Well, we ended up with 6 carbon dioxide molecules that we expired. We ended up with a net gain of 4 ATP, and we also ended up with 10 NADH and 2 FADH, high energy electron carriers. Well, that's a lot of steps for only 4 ATP. But the cool thing here is that the big payoff comes with those high energy electron carriers, NADH and FADH. That is where most of the potential energy from that glucose molecule is currently stored. And to get at that, what we have to do is move to the final stage of cellular respiration, what we call the electron transport chain and oxidative phosphorylation.